Now in this video I will show you how you can use the concept of electric potential when you work with biological molecules. And in this case I'm gonna give you the example of how you can use potential when you work with DNA. It is possible and also very useful sometimes to think about how you can use the concept of the electric potential and the electric field when you work with proteins and enzymes in particular. I will talk about that later. Hopefully I will remember. Now what I have here, this is I'm using a program, UCSF, the University of California, San Francisco. Their program is called UCSF Chimera. It's one of the computational research groups there has developed this tool that you can use to visualize molecules. It is free. You can download the program for free. Now, what I did, and if you want, you can do the same thing. You can go to File, Open, Fetch by ID. The one that I did was 127D. Okay, if you do this and then you click on Fetch, you're gonna see the same thing that I see on my monitor now. Now, what is this? Of course, so it looks like a double helix. It is a right-handed double helix. And now, in addition to the double helix, you can also see that there is a small molecule on the minor groove of this DNA molecule. So here, this large area, that's your major groove. That's your major groove. This area here, this volume here, where you see it's the two backbones are very close to each other, or closer than what they are on the major groove. This is how we define the minor groove. Okay, and you can see here that small molecule. Let me see. I may be able to. So, okay, what you see here with green, this is the small molecule I'm talking about, just to make it clear. Now, I will select the DNA molecule and I want to show the surface. Okay, now there are I want to talk about the representations. There are a number of ways that you can use to represent the same thing. At the end of the day, everything is a model. Okay, the DNA molecule in reality is the DNA molecule. Okay, you have the electrons moving around, their wave functions, the electron density. You have the nuclei with their neutrons and protons, and so on. We can, though, represent the same thing in different ways. And why are we doing that? Well, because each representation is helping us in a different way. So there are, when I want to think about how small molecules bind to DNA, often, not always, often, the most useful way to think about it is to visualize the DNA molecule in terms of its surface. Now, this is, okay, if you think about the volume that the electrons of the atoms that make up my DNA molecule, if I think about that volume, then this is what this surface is showing me. Okay, this is, if you want, a model of the volume that the DNA molecule takes. Now, can you really talk about volume when you talk about atoms? Is there when you have electrons, and the way that we describe electrons is with their wave functions and the electron density, it is not really that clear what is the volume of an atom. But in any case, we do have we can use some convention, and a reasonable one that most people agree, and then you have an idea of what the volume is of each atom of this molecule. And if you combine all of those volumes from all of those atoms, you get something like this. Now, so as far as you can see here, everything is just the same boring gray, or I don't know, whatever color this is, white, yellow, but everything, it's the same. Okay, I can tell this is the backbone, this is the oxygen, another oxygen of the phosphate group here, another oxygen, and so on, and the same thing, oxygen from the phosphate group, and so on. That's the backbone. And of course, you can continue to see the small molecule with a different representation. Now, what I can do though with this tool, let's just see. Let's see if I do it this way. Okay, I need to use not this tool. There are a number of options here. Now, what I'm doing, okay, ESP stands for electrostatic potential. And now this one is a little bit more interesting, but the color coding is not great. So let me change that. I do know that it is the DNA molecule has a negative charge on the backbone. It is highly negatively charged. So I'm gonna say, let's say that this is minus, let's do minus 7.5. And I'm gonna set the blue to be at 0. This may make coloring a little bit better. Okay, it seems that, let's do it minus 20. About minus 5. Okay, that's a little bit better. So you can play and you can use more than 3 values, okay, more than 3 colors. At the end of the day, use the color coding scheme that makes you get the most insight from this. And this is what I want to see here. Now, you can see the red corresponds to the most negative value of the electrostatic potential. Remember, on the volume of the DNA molecule, what I'm doing here, on the volume of the DNA molecule, I am showing what is the value of the electrostatic potential. And with red, I'm showing the places on the volume of the DNA molecule where the potential is the most negative. With white, I show the ones that are a little bit more positive, and with blue, where it is even more positive. Now you can tell what we see here, okay? Remember, red is the most negative potential. So here is where I have the most negative potential. Let's spin it around and see what's going on around it. So I see white, a little bit of red here and there, but mainly blue on the ends, and then white on the major groove, and a little bit higher values here where you see the blue. But if you see here in the space, in the minor groove of the DNA molecule around the center, this is where I see the brightest red. That corresponds to the most negative values of the electrostatic potential. Now, I said the DNA molecules are highly negatively charged. Every phosphate group has a charge of -1 because it lost a proton. The structure, the way that the structure is of this double helix makes it so that the potential gets the most negative values here at the center of the DNA molecule on the minor groove. Now, I also want you to keep in mind a lot of the molecules that actually bind to DNA have a positive charge. It does make sense that they have a positive charge because the DNA, the backbone of the DNA is very negatively charged. And you know, a strong attractive force between two molecules in nature will be the electrostatic force. If the two molecules have the opposite charge, they will have a fairly strong electrostatic attraction. Now, how can I use the concept of the electrostatic potential in this case? I know, okay, let's bring up, okay, I know that the change in the electric potential energy is Q delta V. in general, if you keep the charge the same, the greater the delta V is, like in this case, the most negative area, the greater in absolute value the energy that you will have, the potential energy you will have in that area. So when the small molecule binds in that in the area where the potential is the most negative, this is one way to maximize the interaction by maximizing the potential energy, okay, or the absolute value of the potential energy. Now, this is one way to think about how can I use the concept of potential when I work with biological molecules in biology. So if you want to make a really quick prediction and you know the two molecules that will interact, they have the opposite charge, one of the ways to do it is to look at the surface of your biological macromolecule and color code that surface with electrostatic potential. And when you see where the potential obtains its maximum or minimum, depending on what kind of molecule you have, whether it's positive or negatively charged, that will give you an idea of where the electrostatic interaction will be the greatest between the molecule that binds to your biological macromolecule. Now, it is important to keep in mind, okay, the electrostatic potential will not tell you the whole story. It gives you an idea, okay, it gives you an idea of this electrostatic interaction, but it is important to keep in mind electrostatic forces are not the only ones that take place when molecules interact. You do have hydrogen bonds, you do have van der Waals interactions, you do have the presence of the water molecules around them that make things much more complicated. So it's not the whole story, but sometimes it is helpful for us to think about using the electrostatic potential to get insight for the interactions.